Alright, so after a very short first section on our chapter on DNA and gene expression, we're going to move into now our second section. And we have been talking about a couple of other old scientific studies, the Hershey-Chase experiment and the Griffith experiment. There were several other people and researchers involved with the uncovery of DNA as the material of heredity. Rosalind Franklin, we see on the top here of this slide, was instrumental in figuring out the overall shape of DNA as a double helix. She worked with X-ray crystallography and was able to, based on that process and some very complicated mathematical equations, figure out what the shape was. Other people, Shawgraff, Watson, Crick, there's a whole slew of other people also involved with figuring out what DNA looks like and what it does. So we know that DNA is a nucleic acid. We know from back in chapter 2 on chemistry that nucleic acids are polymers that are made up of monomers called nucleotides. And so nucleotides are the building blocks we need to create nucleic acids. And other terms we've talked about before, things like dehydration synthesis. If we take two nucleotides, remove a molecule of water, that creates a bond, in this case a phosphodiester bond, between two nucleotides, join them together. That's the beginning of the process of creating a string of nucleotides and eventually a nucleic acid. In the case of DNA, the 5-carbon sugar is deoxyribose, hence the name deoxyribonucleic acid. We have a phosphate group, which is attached to deoxyribose, and then we have one of 4 different nitrogenous bases. And so the combination of a sugar, a phosphate, and a base, a deoxyribose, a phosphate, and one of these 4 bases, constitutes then a nucleotide. And every single nucleotide has 3 parts, a sugar, a phosphate, and a base. And so when we take the free hydroxyl end of the deoxyribose molecule, we can then attach that to a phosphate group, and then that's going to allow us to start joining together nucleotides through dehydration synthesis. So that's for a single chain of nucleic acids. We also know that DNA is a double helix. So there are 2 chains of DNA wrapped around each other to create the overall structure of the nucleic acid. So it turns out in the middle of those 2 chains, there are hydrogen bonds. So between for example, a cytosine and a guanine, these 2 nucleotides here, we see 3 different hydrogen bonds holding those 2 bases together. For adenine and thymine down here, we see 2 hydrogen bonds holding those bases together. So when you consider the fact that this entire chain of DNA might be anywhere from 78 to 238 million nucleotides long, and for each pair of nucleotides we find either 2 or 3 hydrogen bases between them, that's a huge number of hydrogen bonds per molecule of DNA holding this thing together. Right, now, we've talked before a lot in the course already about genes. We've talked about genomes. We said that genes are just recipes that are used to build proteins, and that's a very important definition to know. Genes are bits of information that are used to construct individual proteins. We're going to look at how that works here in the rest of the chapter, but we want to go back and review what that means, right? So the genome then is all the recipes an organism has to tell it how to make all the proteins that it has to make. Okay, so we've seen genes, see genomes. In a second here, we're also going to see chromosomes. Right, now, for a long, long time, I mentioned before, people have known about DNA and protein, but for the longest time, people thought it was actually protein that got passed on from parent to offspring to tell those offspring how to construct their bodies, how to build their proteins, and how to do what they do. The reason why they thought that is because protein is typically much more complex than DNA is. DNA can be a big molecule, but it's not overly complex, whereas proteins can also be big and far more complex because they have a lot more different building blocks. If we jump back a second and we look at what a nucleotide is, a sugar, a phosphate, and a base, the only difference among nucleotides is what that base is. And there are only 4 different bases to choose from. So for all the DNA in the entire living world, as far as we know, all that DNA is made up of one of 4 different nitrogenous bases in each position, and that repeats over and over and over again. There really aren't that many options for subtypes or nucleotides, building blocks to plug into DNA. Whereas with proteins, there are 20 different possible subparts. There's 20 different monomers that are used to build proteins. So proteins are inherently more complex. People just kind of assumed initially that it was the protein stuff that allowed organisms to pass on information about themselves to their offspring, because DNA was seen as being too simple. Now, what they didn't realize was that if you look at not so much how many different nucleotides there are, but the ways that you can rearrange those nucleotides to create new messages, new information, that's how we get the complexity that DNA is able to convey. So DNA is like a language, right? We could have a language made up of, in the case of English, 26 letters, Hawaiian's like 14 letters, binary is only 2 letters, zeros and ones. They don't have to have that many different letters to create a very, very complex language. The English alphabet has 26 letters, but you can create something like 450,000 different words using only 26 letters by rearranging the order of the letters, by changing the length of the words. You can create what amounts almost to an infinite number of words just by changing up the order and the length of the sequence. And DNA is the same way. Even though we only have 4 different nucleotides, by rearranging the length of the chain of nucleotides, rearranging the order of nucleotides, we can create an infinite diversity of genetic information. So for example, if you were to take a very, very tiny piece of DNA, only 10 nucleotides long, there are over 1 million different ways to arrange those 10 nucleotides based on having only 4 different bases. Remember, 4 different nucleotides. Sugar phosphate A, sugar phosphate T, sugar phosphate G, sugar phosphate C. 4 options. But with only 4 options, if you put 10 of them together in a string, there's over 1 million different ways to arrange those. Now, most genes are not that small. Most genes are going to be thousands, if not tens of thousands of nucleotides or longer. When you start getting to that scale of things, you really start to see how diverse this molecule can be. All right, I'll talk about chromosomes for a second. We've seen genes, which are the recipes to build proteins. We've talked about genomes, which is like the whole recipe book that an organism uses to build all of its proteins. The chapters in those recipe books are called chromosomes. Right? So chromosomes chemically are a set of DNA wrapped up around a protein. Now don't let this confuse you. The protein is not information. The protein is there only as organizational material. Right? The important stuff is the DNA. DNA wrapped up around a protein makes up a chromosome. And chromosome number is a very integral part of what a species is. Humans, for example, have 46 chromosomes in our body cells. Not so much our sperm cells and egg cells. Body cells have 46 chromosomes. Horses have 64 chromosomes in their cells, again, all except for sperm cells and egg cells. Fruit flies have like 8 chromosomes, right? So it depends on what species you're in as far as how many chromosomes you have, but it's the same idea. Each chromosome has hundreds, if not thousands, of genes on it. Right? So humans have 46 chromosomes. Typically, you have to have 46, no more and no less. There's a few cases where you can get by having 1 too many or 1 too few chromosomes, but there really aren't that many situations like that. Most of the time, too many chromosomes, especially the really big chromosomes or not enough chromosomes, can be fatal, typically pretty early on in development. All right, so chromatin is the stuff that's what makes up the chromosome itself. Chromatin is a mixture of DNA wrapped up around a protein. The DNA is the informational stuff. The protein is the organizational stuff. The protein is just there to help organize the DNA to make sure it stays organized, not tangled, not knotted, make sure it's in the right form. Right? So chromosomes are a single long piece of DNA. If we were to go back here a second, if we could take a look at one of these chromosomes, say right up here, if we could zoom in all the way on the very, very, very end of it, as far as you can, what you'd see all the way in here would be a DNA double helix. And that DNA double helix is wrapped up very, very tightly back and forth and back and forth and back and forth and back and forth around a protein until eventually, by the time you get down here and zoom in really close here as well, you're going to see again a DNA double helix. So this entire thing here, just this side, is one continuously long piece of DNA with a beginning point and an end point. It's all wrapped up around a protein. Okay, so for an average size human chromosome, it's about 140 million nucleotides long. That's for a medium-sized chromosome. Somewhere around chromosome 8 or chromosome 9, somewhere in that neighborhood, is about 140 million nucleotides. The shortest chromosome in the human genome is the Y chromosome, which is what males have, one of those, which they get from their father. The Y chromosome is only 78 million nucleotides. The biggest chromosome, which is chromosome number 1, is about 238 million nucleotides. These are very, very big molecules. But again, aside from being very large, they're not especially complex. That 140 million nucleotides, there's one of 4 options for each and every one of those nucleotides, either an A, a T, a G, or a C. The other big part about this chapter is gene expression. Gene expression, for all intents and purposes, means transcription and translation. Gene expression means taking DNA, turning it into protein, and the processes you use to do that are transcription and translation. Now, another really important factor to consider is that genes are not expressed all the time. Every single one of your cells has exactly the same DNA, with a couple of exceptions. Your gametes, again, sperm cells and egg cells are a bit different. If a cell has a mutation, that might be a bit different. But other than that, all the cells in your body have exactly the same DNA. Now, how do we know that? Well, where did you come from? You started off as a zygote, single fertilized egg, right? That zygote had a certain set of DNA. What happened to that zygote? It divided. That one cell became 2. And when they divided, they both had exactly the same DNA. Fast forward through that process several thousand more times, Now you're made up of an organism of 100 million cells on average. Each and every one of those 100 million cells has exactly the same DNA. The question is, do they use the same DNA? And the answer, of course, is no. Liver cells use a different subset of genes than brain cells use, and brain cells use a different subset from skin cells, and skin cells use a different set from pancreatic cells. They only use the genes they need to use. They don't use the other ones. They're left off. They're not expressed, in other words. Right, so another way to think about that, and it's kind of ridiculous, but it actually illustrates the point, is that the cells in your tongue actually have all the genes they would need to make hair. Now, clearly, they don't use those genes, hopefully at least, for your sake, I hope they don't use those genes, but they have them. They're there, they're just left off. And as long as they're left off, they're not going to produce the proteins, they're not going to respond, they're not going to look like the kind of cell that normally would produce those proteins. So gene expression is turning DNA into protein. Okay, so DNA stores genetic information. Back in the time of Watson and Crick, in the 1950s, there was this idea called the central dogma. We've talked about this before already in class, but the central dogma says this: DNA flows, information flows into RNA, and RNA information flows into proteins. And really, that's the meaning of life from a biological perspective. There are really very few exceptions to that rule. The only one exception is what's called a retrovirus. Retroviruses are able to go backwards. They can actually take what their genetic material is actually RNA. They can turn it into DNA by using an enzyme called reverse transcriptase. And so when they do that, they're actually really sneaky here. They take their RNA and they actually turn it into DNA inside one of our chromosomes, and it stays there, maybe indefinitely in some cases. So it's a way to go backwards in the central dogma. But without that exception, everybody else goes DNA to RNA, RNA to protein. The DNA to RNA process is transcription. The RNA to protein process is translation. And that's mostly what the rest of this chapter is about, is how transcription work and how translation work. These two processes together, again, are called gene expression. Okay, so this is a very important topic. Now, a little bit more on transcription and translation. We said transcription is the conversion of DNA to RNA. DNA is used as a template or a set of instructions on how to build RNA. This typically only uses one strand of the DNA template. The other strand is not used. A few differences here between DNA and RNA throughout the process is DNA contains deoxyribose, whereas RNA contains ribose. That's one important difference. The sugars are different in DNA versus RNA. DNA uses ACGT, RNA uses ACGU. So when you're going from DNA to RNA, anywhere where you would normally plug in a T, you have to plug in a uracil or a U instead. And then also DNA is a double-stranded molecule, whereas RNA is only a single-stranded molecule. So there's some differences here that we have to kind of accommodate as we're going through transcription. There are different types of RNA produced as well. The most important one that we're going to talk about is messenger RNA, or mRNA, which is used as a middleman of sorts to carry information from the nucleus to the cytoplasm, specifically to a ribosome, where the proteins can actually be built. We'll see some other types of RNA as well, tRNA and rRNA especially. They're involved in translation more, but they're also types of RNA that are also transcribed from genes. Translation is the actual building of the polypeptides or proteins. Remember, polypeptide and protein are the same thing. You can use those interchangeably. A polypeptide is a molecule which has an abundance of peptide bonds, which are what you find between amino acids, so therefore it is a protein. This happens, of course, at the ribosome. Ribosomes can be either free in the cytoplasm or attached to a rough ER, as we saw back in our chapter 3. This is where the mRNA, tRNA, and rRNA are all going to come together to actually form a protein. Right? So the intermediaries, rRNA, tRNA, and mRNA, are needed because the DNA, the genetic material, the instructions for protein production are kept in the nucleus, at least in a eukaryotic cell, and the machinery you need to build a protein is kept out in the cytoplasm somewhere. So we have to get information from point A to point B without removing the DNA from the nucleus, which is kind of key. So this is going to require some middleman molecules. The mRNA, like I said, carries the message of DNA. The rRNA is part of the ribosome. rRNA stands for ribosomal RNA, and it's actually physically part of the ribosome itself, as we'll see. And tRNA is transfer RNA. tRNA is the one that's actually out gathering up amino acids, bringing those to the ribosome where they can be plugged in together to form the protein. All right, so we've answered this question already, but just to go through it again quickly, DNA versus RNA. DNA contains deoxyribose, RNA contains ribose. DNA is double-stranded, RNA is single-stranded. DNA has the bases ACGT, RNA has the bases ACGU. So some very important differences there. And this table kind of compares the two. The sugar is deoxyribose for DNA, no oxygen here. Just ribose for RNA, where you do have an oxygen or hydroxyl group there. The bases are different. ACGT for DNA, ACGU for RNA, double-stranded for DNA, single-stranded for RNA. And we also talk about some functions that are different as well. Right? So those are kind of our comparisons between DNA and RNA. We talked about the bases as well, the different middlemen molecules as well, the intermediaries, the RNA molecules, which are used to actually create the protein out in the cytoplasm. So in a very loose sense, this picture kind of illustrates what's happening in a cell. Right? We've got DNA turned into RNA in the nucleus. This is transcription. We're going to take this RNA after some processing out to the cytoplasm. So this RNA can go into the cytoplasm where it can find a ribosome. And the ribosome is going to read the RNA, the mRNA in this case, to create a protein by using building blocks. And so down here, we have a set of instructions, cookbook in this case, which is transcribed into a recipe. So we're going to copy one of these recipes here. We're going to send this recipe out to the cytoplasm where ingredients have been gathered up. We're going to put those ingredients together, and we're going to get some final finished product. And that's what translation is. Okay, so the next section we're going to start taking a look at is transcription. So I'm going to shut this off here so I can do transcription as one continuous block. We'll see how DNA is turned into RNA, and then also take a look at how RNA is processed in eukaryotic cells before translation begins.